All right, phylogenies. We've already talked about phylogenetics and how we can organize and separate species based on their derived characteristics. One of those, and the simplest way to do that, is through their genetics, but there are other ways to do it as well. So we're going to look at phylogenies, the different terms associated with them, and different types of phylogenies in this lecture. So first, taxonomy is a very much older discipline than phylogenetics, but phylogenetics at its roots is just a branch of taxonomy, which is the naming and classifying of organisms. Our original taxonomist was Carl Linnaeus. We'll mention him later. Systematics then is the science of classifying organisms by their evolutionary relationships, which may or may not be morphological, right? So we can look at all these beetles and we can name and classify them by their different characteristics that are similar or different. But if we want to look at evolutionary relationships, we want to specifically understand common ancestors, when they diverged as species, and things of that nature. Macro evolutionary events. Cladistics then is grouping organisms based on shared unique characteristics. So we can classify all of these as beetles because they all have the same wing structure, a similar body plan, a similar shell structure, and we can look at their DNA and evolutionary relationships and call them together as a group beetles. Phylogeny then is the evolutionary history of an organism, usually displayed as some sort of tree, a branching tree with each node as a common ancestor. We'll talk about phylogenies as well. So here's our man, Carl Linnaeus. He was the man who developed this binomial nomenclature and hierarchical classification of taxonomy. Binomial means two names. Nomenclature is the naming of things. So when we talk about a species name, we are using our binomial nomenclature, the genus and the species, with the genus capitalized and the species lowercase, both of them underlined. This is called the specific epithet. And every species has a unique species name. All right, here's Puma con color. This is the mountain lion, and we have Homo sapiens above it. Okay, you can also italicize it if you are writing it or typing it. If you're writing it out by hand, then you would just underline. All right. So the specific epithet is the most specific part that every species has that is unique, but we can group out from there to genus, and organisms have shared genuses, all the way up to the most general, which is the kingdom, and actually domain, which would be more general than that. So you can see the further you get down this hierarchical classification, the more restrictive you get in the individuals which are in that. Okay, and to help you remember this, you can use this acronym. Did Katy Perry come over for good spaghetti? She didn't to my house, but that can help you remember domain, kingdom, phylum, class, order, family, genus, species. And if you watch that YouTube video, it's just a silly one where Mom's spaghetti is inserted into a Katy Perry song. So a win-win, really. All right, so a phylogenetic tree then is a phylogeny which has a branching diagram. And it represents the evolutionary history of that organism or the organisms that you're looking at. Mostly matches the hierarchical classification that we talked about before, kingdom, phylum, class, order, but it can split things even more into even finer and smaller and more relationships. There is mismatch that sometimes occur, usually due to derived traits, and DNA similarities have often caused us to reclassify our relationships among species and taxonomic groups. So a phylogeny is, I want to emphasize, a hypothesis. So it is not a fact. It is an organization or representation of what a scientist or someone who has analyzed that information thinks is the best representation of the relationship, but it can change, or you can have different hypotheses for the same group depending on what characters you use. You can come up with a different answer. All right, but essentially every branch has at least two, or every node has at least two branches coming from it. Each branch point or node, you can see that on the figure there, represents the common ancestor and divergence away from it. Sister taxa are those that are at the ends of the branch and they share an immediate common ancestor. Basal taxa are groups that diverged early in evolutionary history. You see that there. The basal taxa on that one came out much further down on the root. And a polytomy then is a branch point where it's unclear. So here you have 4 different branches coming out of the same ancestor. Now, generally that's not considered to be very possible, very unlikely. And so it's more likely that the divergence between those 4 species does have a clear common ancestor, and 2 would be parsed out at different times. But we don't know, and so we represent it as a polytomy, an unknown branch point. All right, so here's a phylogeny of Mysticete whales, one that I used for my dissertation. And you can, one of the things we'll do in class and possibly on a test is identify different characters on here. All right, so do you see any branch points? Yeah, there's lots of them. There's one there between the brides and the sea whales. There's one there between different groups as well. Sister taxa, all right? Northern and southern right whale are grouped together as the fin whale and the Antarctic minke whale. Actually, no, those are not sister taxa. Even though they're next to each other on the phylogeny, the Arctic minke whale would be a sister taxa to the North Atlantic minke whale. Are there any basal taxa? Yes. We have a sperm whale here who's all by himself compared to the rest, and we have a hippopotamus who's all by himself compared to the rest. So they diverged much earlier than the rest, and in fact these are not Mysticete whales, and so they would just be used for comparison. Do we have a polytomy? Yes. Look here, we have 1, 2, 3, 4 branch points coming out of one node. We don't know how this diverged based on the information given. All right, so to construct a phylogeny, you can do this based on morphological characters. You can also do it based on DNA, RNA, proteins, molecular characters. And homologies then are the characters which are similar due to shared ancestry. And so we can use those to group organisms and consider where they diverged. Homoplasies, on the other hand, are characters due to convergent evolution. So they're analogous traits, and they are actually not helpful and often will confuse the phylogenies which are produced. All right, again, cladistics, we're looking at how we can group organisms often on our phylogenies. A clade is a representation of a group of an ancestral species and its descendants, and there are 3 different types. A monophyletic clade means you have a descendant and all its ancestors. So this yellow one here, if you could trace the node from the New World monkeys all the way back to where it branches to the rest, you can see that that node and all of its descendants are included. And so that would be a monophyletic group. Paraphyletic group is where it contains some but not all of its descendants. So you go back to this blue one, you can see the node between the lemurs and lorises and trace that back to where it comes to the rest of the group and follow that, and you see that the blue is not circling all the ancestors. It's excluding the yellow guys. And so that would be a paraphyletic grouping. A polyphyletic grouping is one that just grabs 2 different things and puts them together. So let's say lorises and tarsios have really big eyes, and so we're going to put them together for a group. Well, that does not reflect an evolutionary relationship, so they have different ancestors, and it would be polyphyletic. All right, so what would you call this grouping here? I circle all of these, monotremes, marsupials, and eutherians, and stretch it back all the way to the common ancestor of them. This would then be a monophyletic grouping. All right, if I did this one where I said reptiles, turtles, lizards, snakes, and crocodiles, well, that actually excludes birds, so it would be a paraphyletic clade. So the birds actually share a common ancestor with crocodiles, and so the reptile clade is paraphyletic. All right, as we mentioned, shared traits are important in these phylogenies. Shared derived characters are those that are unique to their clade. A shared ancestral character is one that is not unique and is shared to the basal with the basal taxa generally. And if we can parse these out, those that are derived or unique and those that are shared, we can then use those to make our phylogeny. So here's an example. We have outgroup C, right, in this first phylogeny. An outgroup is one that is used for comparison, and it's our basal group generally. So let's say A and B have a long tail, and so does C. Well, we can then infer that, well, having a long tail is a shared character. And so it's not one that's going to help us understand the difference between the two. Well, let's say A and B have sharp teeth and C does not. Well, then we could say, all right, well, that must be a derived character shared by A and B, which is different from the outgroup. So in-group is the taxa being studied, and the outgroup is the one used for comparison. And I just basically did that comparison to show how we could use characters to do that. All right, so this bottom phylogeny, if we were interested in the relationship between Hylobates and Symphylonegus and Numascus genuses, we would use the top two, Pongo, as an outgroup, and then able to make inferences based on those. So there are actually different ways that we can make phylogenies, lots of different ways, depending on how many individuals we have and where we put the branch and things. But the best one is generally the one that is the simplest, the one that has the least number of characters. Okay, and so you can see on here, if I say red feathers evolved twice, that puts on 2 characters. If I move it further back and say, oh, that must be actually a shared derived character which only applied once, that would be more parsimonious, less complicated, more simple, and thus more likely. We'll do some of this in class. All right, so we're going to briefly talk about the universal phylogeny. This is something that has continually been refined over the years. Back in Linnaeus, he just had 2 kingdoms, plants and animals, because he discovered this before bacteria. That was then added in. In the 1960s, we had 5 kingdoms. And then molecular sequencing showed, oh, we have actually some groups which need to be split up even more. And so now we added a group. We added domains in. Now we have domains, which is more general than kingdoms. Our 3 domains are eubacteria, archaebacteria, and eukarya. Eukarya are eukaryotes. We learned about those last year. Both archaea and eubacteria are bacterial clades, and that's it. Had their relationships, this would be the kind of tree of life relationship. But we also have other proposed relationships where it's more of a ring of life, that in fact there wasn't a common ancestor, but of which these diverge, but actually there was sharing of genes between these 3 groups or at least these 2 groups, until divergence came later. And so that would be this ring of life concept. We also have this web of life concept, which you can see in the bottom figure, where there was some crossing, and that also takes into account the symbiotic relationship. I just finished.